We're going to spend some series of lectures on the introduction to clinical trials. My name is DeMets. I'm professor of biostatistics and statistics. I'm in the Department of Biostatistics and Medical Informatics. Even though I was trained in statistics and mathematics and computer science, I spent most of my career in the design and conduct and analysis of clinical trials in a variety of disciplines: cardiology, oncology, AIDS research, and so forth. We're going to be engaging in a series of lectures on the fundamentals of clinical trials. There will be a series of 8 lectures. The first lecture will cover the background of a clinical trial, the history of it, some of the ethical issues, the regulatory issues, as well as local concerns that must be addressed. We'll then go on to why we do clinical trials and where the clinical trial methodology fits into the general clinical research enterprise. One of the more challenging things of a clinical trial is to figure out what the question is. And so defining the question will be a topic of one of our lectures. After that question has been defined, we will then look at the basic trial design that will be most appropriate and efficient to answer the question you've posed. Every clinical trial needs to decide how large it must be. So we'll examine some of the key issues in determining the sample size to be estimated for your trial. Every trial now also must have a monitoring plan. So we will examine some of the statistical methods, and just in principle, that are used for monitoring a trial for both safety and benefit, as well as some of the ethical concerns that must be kept in mind. After we examine monitoring a trial, we will then look at the issues that must be kept in mind in analyzing the data. Not the statistical techniques, but more the principles and philosophies. And finally, we'll look at and examine the amount of data and how it should be reported, both in the literature as well as to the medical community. By looking at all of these issues, we hope that you'll better be able to review the clinical trial literature and begin to have some of the background, perhaps to design your own study. I've given you some references here that you can look at if you wish to get more detail on any of these topics. These are all authors who have made major contributions to the field of clinical trials, and you'll find a lot of material of interest in those texts. Regardless of what our careers are, most of us will be reading the literature, either to further our clinical practice or to follow recent advances in clinical research. In either case, we need to be critical reviewers of the literature throughout our careers. Some of us, including myself, are engaged in the conduct of clinical research, and so we need to understand some issues and key principles in the study design and analysis. Typically, the results of our clinical research are published in leading journals. We need to understand that not all articles published are of the same quality, regardless of the prestige of the journal. Furthermore, not all evidence in the journals are of the same level. There may be anecdotal data, which is important, maybe observational studies, or maybe experimental studies. Each of them give different levels of confidence of the data. And as practitioners and researchers, we need to sort out that evidence and how we should use it. We're going to start with some basic background and history of clinical research to sort of set the stage of how we operate in today's environment. Research cycles include at least 3 areas, which is an oversimplification, but we can start with basic research in the bench or animal research. We may have observational research, which includes observing large cohorts of individuals, either those with disease or those who are healthy. We may observe or try to tease out what risk factors are the cause of these disease incidents, go back to the bench and find ways to modify those risk factors. But eventually we have to translate that research into a clinical trial or a clinical research environment to find out, does it work? So there's no one piece is more important than the other. All 3 are complementary. All 3 are necessary. We're going to focus our energy on the discipline of clinical research. It's rather interesting from my perspective and the different disciplines that come together to form a common core of knowledge. The clinician, of course, is critical because they're the ones who are delivering the therapy or the intervention. The statistician usually contributes to the experimental design, the data analysis, and a lot about the monitoring of the trial as it goes along. The behavioral scientist may be important in modifying patient behavior or individual behavior, or in evaluating the outcomes of the studies we do. Clinical pharmacologists are important if we're doing a drug study. And then there's a discipline which is evolving or emerging as the clinical trialist, the individual who focuses on how to actually pull off or conduct clinical trials. Historically, we trained our current clinical research workforce, a multidisciplinary workforce, by sort of on-the-job training, what I call a trial and error. As we learned as we went, we made errors, we corrected those errors, and we moved on. Only now are we beginning to develop rigorous training programs in clinical research. And this is important for reasons which I'll get to. So there are many disciplines now involved in clinical research that really didn't have formal training but came up through the on-the-job training. Another aspect which we have to think about is that there's the aging of our current clinical trial researchers. And if we don't begin to train the next generation, in a few short years, within a decade, we will be having a serious crisis shortage of those engaged in clinical research. To back that up a little bit, I've got some data here. You can see it in the above graph. It's plotting by year the number of physicians going into patient care, into research, and teaching. You'll notice that there's been a continual increase in physicians going into patient care, but no increase whatsoever in those going into teaching and or research. This has been constant for a long time, the past couple of decades. Furthermore, if you look at the aging of the National Institutes of Health funded investigators over a couple of decades, you can see that the percent of investigators who are over the age of 50 has been increasing. In fact, now the average age of a funded investigator is in the mid-50s. So we have an aging of our clinical trials research workforce, and we need to do something about that. This series of lectures is trying to help that process. The clinic is a perfectly good laboratory. Clinicians in their busy practice can also participate and lead research efforts. In today's environment, it's very difficult to be in the clinic mostly, a lot of pressure to be in the clinic these days, and still run a competitive wet lab research activity. The types of clinical research that we do range from the case report, the anecdotal incidents, which identifies a problem. We do engage in lots of observational studies. We observe patients who have a disease and try to understand what their background, what their exposure was, and compare that to a healthy population. We call that the case control or retrospective design. Much of the work on the causes of lung cancer, i.e. smoking, were developed through such a design. We can do cross-sectional studies where we measure a population at one point in time. I've given two examples here. The WESDR is a cohort of diabetics that were in southwestern Wisconsin that were examined over a short period of time, and also a study in Beaver Dam, Wisconsin that we've examined for eye disease. But it's one snapshot in time. We also engage in prospective studies. We identify a cohort, perhaps initially as a cross-sectional study, but then we follow them forward in time. The most famous of these is the Framingham Heart Study, which identified such now commonly known risk factors as blood pressure, cholesterol levels, diabetes, obesity. The WESDR-2 was the follow-up of the initial cross-sectional cohort in southwestern Wisconsin. These studies identify associations, high cholesterol associated with incidence of heart disease, and so forth. Then we have drug development, or you could say device development as well. You go through various phases. Phase 0 tends to be animal lab studies. Phase 1 is the sort of first in human studies. Phase 2 is studies that we'll talk about where you try to identify, does the device, drug, biologic have the activity that you want it to do? Does it lower blood pressure? Does it lower cholesterol? And finally, we have the experimental phase 3 where we're really doing comparative studies to find out whether the therapy actually works in the way we want it to. And there's various ways to identify the control arm, the comparative arm, historically concurrent and randomized. We will look at those in detail later. But these studies are trying to understand the effectiveness of the therapy or the intervention that you have in mind. We believe in the world of evidence-based medicine that the randomized clinical trial is the gold standard because it minimizes bias. That's a very important issue, in fact, maybe the most important issue in conducting experimental research. We can't do randomized clinical trials for all questions. We don't have the time or the funding, and sometimes it's not ethically permissible. We must make choices on what evidence we will use to develop our clinical guidelines. Recent history teaches us that we must be cautious in the use of our evidence. If we don't seek the most rigorous evidence that's available, we can develop problems in our clinical practice and research. Theory and observational studies based on evidence have not always led us to the correct conclusions for very important questions. We'll see some examples of that as we go. If you're in clinical practice, you're quite aware of guidelines. Guidelines have become common in many of the disciplines. which sort of identify for certain clinical conditions what the preferred therapy is. Those guidelines, hopefully, are based on evidence, and as I indicated earlier, there is different degrees of evidence. We have come to identify different classes of recommendations. Here is one example. Class 1 is that the intervention is useful and effective based on probably randomized controlled clinical trials. Class 2 is that these are mixed opinions. There's some evidence that the therapy is effective, but there's also some evidence that's to the contrary. We call that 2A. 2B is evidence that differs also, but it tends to lean against, that is, it's not in favor of efficacy. And finally, Class 3 is that the intervention is not useful or effective, in fact, it may even be harmful. So we have different classes of recommendations that are sometimes given to evidence. This is one such scoring system. Sometimes medical societies come up with other strengths of evidence. Here is one that the Heart Failure Society of America came up with. Level A is that it was based on randomized controlled clinical trials, a single trial or a series of studies. Strength B is it was based on observational studies, either cohort prospective studies or case control studies, or perhaps it was part of a subgroup of an earlier study. And C is the expert opinion of a group of individuals interpreting what data does exist. What's the alternative? Well, there's all kinds of alternatives. Probably none of them are optimal. There's evidence based on the prestige of the individual who's promoting the idea. It may be on the eloquence of the individual, strength of his arguments, or it was the last patient-based evidence. There's many such ones that you can read in the British Medical Journal, authors that have talked about this type of evidence. It's not the kind of evidence we have a lot of confidence in, and so we probably want to move away from that. You should also be aware that while we have historically believed that clinical trials were the way to go to understand evidence, there's lots of challenges to clinical trials. There's been congressional legislation, which I'll refer to in a minute. Even the leadership of the National Institute of Health and the Food and Drug Administration have challenged the utility of some of our clinical trials. There's been pressures from Wall Street and the Wall Street Journal. Patient advocacy groups have been challenging why do we need clinical trials, why can't we use some of these alternative evidence-based medicine methodologies. In the proposed legislation, for example, there was 3 tiers that were proposed. Tier 1 was that you could approve new therapies on phase 1 information, that is first-in-man studies, and it should be based on clinical but not statistical analysis. Yes, you may require some post-approval studies, but that the early phase information would be sufficient for a Tier 1 approval. Tier 2 is it's based on intermediate outcomes, or what we call surrogates, or some biological marker. Tier 3 is what has been the standard requirements and what I will be speaking mostly in these series of lectures. Furthermore, in this legislation, there was been the challenge of using placebo-controlled studies, that they wanted to de-emphasize the statistical analysis. In fact, said no disapprovals solely on the basis of statistical analysis or confidence intervals. That the evidence could be, in fact, based on uncontrolled studies, such as case histories, observational studies, mechanism of action studies, or computer modeling. And in fact, the outcome data may well be a surrogate or a substitute or some biological marker. We will examine some of those as we go through the lectures. But nevertheless, there's been challenges to something that we believe has been a very useful research tool over the past several decades. What do we mean by a clinical trial? We'll get into the details of this as we go, but for now, it's a prospective study. It's not retrospective. It's in a population of human subjects. The intervention can be quite far-ranging, a preventive therapy, a therapeutic intervention, a diagnostic trial involving a biological, a drug, a device, a procedure, or a behavior modification. The control arm, the comparative arm, might be a no-intervention arm. It could have a placebo. It could be the current standard of therapy or some previous standard, but there's some comparative group. And I said it's in humans, not animals, and that brings in some issues of ethics and informed consent, which we will talk about. Very brief history. If you look at some of the key elements of clinical trials, one of the first planned experiments was in 1721, the smallpox experiment. You could not do this study today for ethical reasons, but inmates in a prison in Great Britain were offered the opportunity to have their sentence commuted if they volunteered for inoculation. All 3 were exposed to smallpox. All 3 remained free, indicating that the inoculation was effective. There was no concurrent control group, but this was a deliberate, planned experiment. Another step in history was the use of a control group. The so-called scurvy experiment of Lind in 1747, they used a control group of sailors who were on board a ship called the Salisbury. Scurvy was a problem. He identified 12 patients, i.e. with scurvy, and gave them in a deliberate fashion 6 treatments. 2 subjects got each treatment. 1 treatment was oranges and lemons. In fact, those 2 men recovered. The others did not. So here was a comparative group. Those who got the oranges and lemons and those who did not, it can make a comparison. The concept of randomization was introduced into the experimental design world by Sir Ronald Fisher in the field of agriculture in 1926. He developed a very elaborate system of experimental design, and this was used throughout agriculture, but it was not adopted in human research or in clinical research until 1931 in a tuberculosis study. It was the first randomized trial, at least that's been identified by some authors. However, the use of randomization in clinical trials really did not get attention in a serious way until 1944 and the late 1940s, when the Medical Research Council of the United Kingdom began to use this design in various studies. The one that most of us looked to was the tuberculosis trial in 1948. It was randomized, comparing streptomycin versus placebo. It was based largely on the work of Bradford Hill, who was one of the authors of the references I gave earlier, sort of the founder of the modern-day clinical trial. But it certainly supported the concept of randomization in clinical trials, which we used to this day. I want to focus a little bit now on the ethics involved in clinical trials and give you some background and some history of the regulations which we all must be aware of in conducting clinical trials, and also the ethical considerations which clearly all have to take into account. Ethics is a very complicated subject. It's the moral quality of a course of action. It has rules that govern the conduct of a profession, and society's views, in fact, can change over time and may differ from one ethnic group to another or one country to another. So there's a changing phenomena. What is ethical today may not be ethical tomorrow, and so forth. So it's something that is very challenging in the history of clinical trials. We need to keep in mind some historical events that have taken place that have set the standards for how we conduct clinical trials. These range from the Nuremberg Code from 1947, the Declaration of Helsinki, federal guidelines, FDA guidelines, and policies which are at work at each of our local institutions. The Nuremberg Code came from the trials that followed World War II, when prisoners of war in German concentration camps were subjected to some pretty atrocious human experiments. The principles that came out of those trials were that patients involved in clinical trials must voluntarily consent to these studies, that the experiments must yield results that are for the good of society, that the experiments have to be based on something, some animal experiments, laboratory research, and general knowledge of the natural history of this disease. All attempts should be made to avoid all unnecessary physical, mental suffering and injury. No experiment should be conducted if there's a reason to believe that death or a disabling injury would occur. That the degree of risk that a patient is exposed to should never exceed the importance of the problem to be solved. That the experiment should be conducted only by scientifically qualified persons. That the human subject involved in the study has the liberty to bring the experiment to the end. They have the right to get out. And that the scientists in charge of the experiment must be prepared to terminate the experiment if the probable cause of continuation is likely to cause injury, disability, or death. We will get into that in some detail later as we examine the process of monitoring trials for benefit and risk. The Declaration of Helsinki incorporated many of these Nuremberg principles and formalized it in 1964. This declaration has been modified and updated over time. The most recent modification addresses the use of placebo controls when a proven therapy exists. The U.S. government convened a conference at Belmont, became known as the Belmont Report, on ethical principles and guidelines for trials or studies which are sponsored by the NIH. 3 principles, I think, came out of that conference. One was entitled the respect for persons, that individuals with diminished autonomy are entitled to protection. such as children, prisoners, those with mental incapacity. The issue of beneficence, that you want to maximize the possible benefits and minimize the possible risks and harm. And the third principle was the principle of justice, that there should be some fairness in the distribution and access to the experimental treatments. That is, giving this to your friends and neighbors would not be considered a fair way to distribute or make the opportunity to participate in a study. There's a large series of Code of Federal Regulations, the so-called CRF series. I've just highlighted a few things which are in there, but you need to be familiar with these Code of Regulations if you're going to be involved in clinical trials. Some of the issues that are mentioned are that risks to subjects are minimized by using procedures which are consistent with good, sound research design and do not unnecessarily expose subjects to risk. That the research plan makes adequate provision for monitoring data collected to ensure safety of subject. Again, the issue of monitoring. We'll come back to that later. And there's adequate provisions to protect the privacy of subjects and maintain confidentiality of data, the so-called new HIPAA rules, which we'll talk about. The Food and Drug Administration is the federal agency that's charged with the approval of new drugs, biologics, and devices for commercial use. The FDA was created in 1906 as the Pure Food and Drug Act, largely to address food safety in this country, but over time it added issues that involve the regulation of drugs, biologics, and devices. These three areas, by the way, have different regulations and different standards because they were added at different times in history, so the standards changed over time. Nevertheless, these guidelines are in the same spirit as the Health and Human Services regulations regarding the standards of approval. We also have another federal office called the Office of Human Research Protection, or OHRP. In some ways, it has ultimate authority about research that's conducted in our institutions. It issues requirements and policies. It reviews our institution review boards for compliance to these policies. And in fact, it may shut down research at an institution for non-compliance to these policies. There have been several cases in recent years where institutions were shut down for research because documentation of consent or documentation or review of protocols was not in compliance with these regulations. At the local level, we have something which is called Institution Review Board, which is required by OHRP. You cannot conduct research without such an Institution Review Board. And this board is charged with review of each new protocol for its merit, the ethics of it, the consent process and its documentation, and it must review each trial annually and also review the safety profile of the studies to assure that the patient safety is being adequately taken care of. The informed consent process, according to the Code of Federal Regulations, must contain certain elements. It concludes that the effective consent must be obtained from the subject or their legally authorized representative. The investigators may exert no coercion or arm twisting. The information must be understandable, presented in a language and a level of language that is commensurate with the population being addressed. The consent must not only be written, but an oral, if at all possible. In the consent document, you must explain the purpose of the research study, how long it will take, and what kind of procedures are going to be involved. You need to identify possible risks. You also should identify possible benefits, not only to the patient exposed to this research, but to other individuals. You must disclose what the alternatives are to being in this study, what other therapies are available to the patient. You need to discuss the confidentiality of the data, also compensation to the patient in case there's an injury. You have to describe what the terms are. You need to identify who the patient should contact for questions or problems or injury. And finally, it must be made clear the participation is totally voluntary and refusal to participate would in no way affect their care. There's some additional elements that are required, especially if there are risks to fetus, but the investigator himself or herself may choose to terminate the study for that patient. There may be some additional costs that need to be identified. That if the patient should withdraw from the study, what are the consequences of that withdrawal? That the results of this research will be shared at the end of the study with others in the research community, but also new findings during the study, such as new toxicities that were not known prior to this study, will be shared with the patient. And also identify how many subjects are expected to be entered into this study. How large a study is this? How long will it take? In some exceptions, you may have to get a consent waiver. For example, if you're doing emergency room research, where presumably the patient would not be in any position to provide consent. To do so, however, requires certain procedures. There must be an approval by the local authorities, the local government, or the local environment. You have to establish there's no other way to carry out the research, and that the research involves no more than minimal risk. That is, the patient would not be put at any more risk by being in this study than they would otherwise, given their condition, and that the waiver does not adversely affect the subject's rights or their welfare. This is not done very often, but it's most often done in examining, I said, emergency research questions. There have been some recent developments which also affect the way we conduct research. The Shelby Amendment in 1998 is rather important because it requires that federally funded researchers, you and I, that if we conduct research, we must make the raw data available that supports the results of that research. If it was funded by the federal government, we must share the results. Now, this may require effort to document the data. It raises the possibility that researchers could be scooped by our own data. That is, someone might find some result in our data that we did not appreciate and publish that. It may generate many secondary analysis that others might publish. It challenges the privacy of medical records. And the consent forms may warn that privacy cannot be guaranteed. This is in conflict, of course, with the HIPAA rules. Other recent developments is the U.S. National Bioethics Advisory Commission commented that recent recommendations for research abroad, something that's going on more and more, should provide established effective treatment to all participants in the study, whether or not it would be available in that country. An exception could be made if providing treatment would be rendered irrelevant in the host country. But in general, this is a very challenging recommendation. And then, as a follow-up to that, how will the treatments, if they're proven to be successful, be made available to research participants in that country as a whole? Other recent developments is that the Declaration of Helsinki was modified in the year 2000 and examined the question of placebos used in clinical trials when no other therapies are available for comparison. The general idea is that new therapy should be tested against the best current therapy. This suggests investigators divulge to patients also how their trial is funded and potential conflicts of interest. Conflicts of interest have gotten a lot of attention in recent years, and so it's an issue that the Helsinki Declaration examined. Finally, a very important thing is that all trials, all study results, should be published whether the results are positive, that is beneficial, or negative, i.e. harmful. Regardless of the results, they should be made available to the medical community and to the public at large. Another event in history was the death of a gene transfer patient at the University of Pennsylvania. There was a lot of issues as to how this study was monitored and when this death occurred, who was made aware of the death, and the whole monitoring process. Secretary of Health and Human Services, Donna Shalala, got involved in this project, this issue, and in a New England Journal article, a press release, as well as in an executive order, commented that institution review boards are not often provided sufficient information to evaluate a clinical trial completely for benefit and safety. As a result of that, the NIH now requires monitoring plans for all of their trials, Phase 1, Phase 2, and Phase 3. And later, the FDA issued guidelines for data and safety monitoring boards and IRBs. We'll examine those again in detail in a few lectures. Another issue that we have to be aware of is the Health Insurance Portability and Accountability Act. This effort began in the early 2000s as a result of a couple of events. One was that the university had 4,000 patient records hacked. Another was that organ donors had their identity released to recipients. This invasion of patient privacy caused great concern about this issue. However, there's a need for health care reasons, as individuals and patients move across state lines, to be some standardization. And also there was a great desire to avoid employer and insurance discrimination by our health information. So the HIPAA laws require that we have workforce training. Then we notify patients about the confidentiality and the use of their data. That access to data is on a need-to-know or a need-to-use basis. That the research data should be limited to what is actually needed. As I indicate, not a bad idea. We have a tendency to collect too much data, much of which we don't use. And also a need to upgrade data security, which is a growing and continuing issue. In the HIPAA regulations, it refers to the concept of public health information. This is data in any form created, received, or held by a covered entity, such as a hospital, a practice plan, a research institution, that relates to the physical or mental health of the patient, provisions of care and payment, and could be used or would identify the individuals. This kind of data is referred to public health information. It does not include blood and tissue specimens, but the information associated with it is viewed as public health information. In order to comply with the HIPAA privacy rules, you have some options. One is to obtain the written patient authorization at the beginning in a separate document, or it can be part of the informed consent document, where the patient authorizes the researcher to use the data under the conditions provided in this authorization. Another option is to apply to your institution review board for a waiver, or you can develop what amounts to a contract for a limited data set and limited use. The final option is that you can develop an argument that there's a low risk of ever being identified statistically. Obtaining a subject authorization says that you must describe to the patient or the participant the public health information to be used and what will be disclosed, to identify who will have access to that data, what the purpose of the disclosure of the data would be for research, for example. Is there an expiration date or is it ongoing? And that the individual has the right to revoke at any time their authorization to use this data. However, the public health information that's gathered prior to that revocation could still be used. Authorization needs to be signed and dated. The second option of getting a waiver, you have to establish research on the existing database will have no further contact with the individual. And the IRB must determine that there's no more than a minimal risk of the privacy. There's a proper plan to use the data. There's a plan to destroy the identifiers, the patient identifiers, and that written assurances that the public health information will not be disclosed to someone else. And finally, that the research cannot be done any other way without this waiver. The limited data use agreement is an option which is a contract essentially between two parties, the covered entity, those who hold the data, and those who want to use the data. It specifies what data is requested and how it's to be used. Identifies what it can be disclosed. It identifies the obligations of the recipient as those who are going to use the data. There's an effective date, how long, and how it's terminated. You must identify the notices and reporting processes and a signature, usually involving legal review. A very restrictive approach. The issues or the options, authorization, the question is, do subjects really understand the authorization form, truly, fully? The waivers, it's easier and safer to say no than yes. So it's not an easy option. The limited data use, the burden is really on the recipient. In your research, you get a new idea from the data you're examining. It requires a new agreement because it doesn't fit in the scope of your initial agreement. And as far as the last option of statistical assurance, I don't think we can ever really assure that because there's a lot of very clever algorithms that can be used in artificial intelligence to identify sources of data. So it would be very challenging to give assurance that the data would never be identified, especially in the days of genomics. However, we all understand that sharing of data is critical. At the end of a study, we want to publish results in a journal. In the journals, however, you publish summary statistics. There's many ideas that you may want to follow, or someone else may want to follow, that requires access to the raw original data. So without access to the data and being able to share it, research can't progress. So it's a goal which we want to achieve, but yet we have to be conscious of these restrictions. The HIPAA laws have a major impact on how we conduct research. I'm sure have not heard all aspects of this yet. For sure, data security is a major topic, and it's something that the clinical trial world, the biostatistical world, must get involved with. Unfortunately, it does open us up for litigation, so we have to be really conscious about complying to the rules and regulations. So we've been covering some of the issues and environment in which clinical trials are conducted. These include some of the ethical principles that we need to follow, some of the federal rules and regulations which we need to be conscious of, and some of the history of the key components of trial design and conduct. In future lectures, we'll be examining some of these issues in more detail, and stay tuned for the next series.